To begin muscle contraction, you must first know the different parts and how they connect. This is a motor neuron, which connects to a muscle fiber by the synaptic cleft. The synaptic cleft is on the membrane of the muscle fiber, or this black line, also called the sarcolemma. As an action potential moves down the motor neuron, it reaches the axon terminal. Here, voltage-gated calcium channels open, allowing calcium ions to flow into the neuron, which then bind to vesicles of acetylcholine and push it towards the membrane. After it diffuses into the synaptic cleft, the acetylcholine binds to nicotinic acetylcholine receptors and voltage-gated sodium channels, which allows sodium to enter the muscle fiber. As the ligand-gated ion channels open, it triggers an end plate potential along the motor end plate. It then travels down the T-tubule, which releases chemicals. These two chemicals travel to the sarcoplasmic reticulum, where they bind to dihydropyridine and ryanodine receptors. This triggers calcium release by the sarcoplasmic reticulum. This is one single muscle fiber. One muscle fiber is made up of many myofibrils, which is made up of sarcomeres. This little portion here is one sarcomere. One sarcomere is made up of actin and myosin. Actin is the thin filament, which is made up of actin, tropomyosin, and troponin.
Myosin is the thick filament and contains many myosin heads.
When the calcium from the sarcoplasmic reticulum bind to the troponin on the actin, the troponin and tropomyosin move. But for the purpose of this project, I'm going to remove this part, although it does not actually come off. This reveals the active sites on the actin, which the myosin heads then bond to. When ATP, or adenosine triphosphate, is introduced to the myosin head, it cocks it back. This movement is called a power stroke. After the ATP binds to the myosin, it loses a phosphate, creating ADP, which then leaves. When the active sites on the actin are exposed and the myosin heads fit into them, it is called a cross bridge. Once a cross bridge is formed, the actin and myosin slide past one another. As the actin and myosin slide past one another, the myosin heads get stuck on the actin. It cannot leave until a new ATP comes along and breaks the bond, allowing it to contract. The close-ups that I showed you are in this drawing, where the actin and myosin slide past one another in a muscle. This entire cycle is called the contraction cycle, and when bending our arm, it helps us form a muscle. Thanks for watching!